So, welcome to the High Resolution Whole Brain Electrophysiology webinar. Speakers for this webinar will include Don Tucker, the CEO, Wei He, the Director of Sales and International Strategy, Baral Sheth, Director of Sales and Business Development, and Justin Nichols, Sales Manager for North America. We ask that you put questions into the chat of the Zoom, and we will answer them during the Q&A at the end of the webinar. And with that, I will go ahead and turn over the screen to Don Tucker to begin the presentation.
Great. Thanks, Corin. Let me share my screen. And let's see if I can get this started. Okay, can you see that?
Yes.
Great. So our goal today really is to go over some basic principles of whole brain electrophysiology and to describe the approach that BEL takes to the essential aspect of this, which is modeling electrical fields in head tissues such as that we can localize from a high density array to characterize the activity of the brain. So BEL started at the University of Oregon a number of years ago, quite a few years ago now. And we created EGI in order to commercialize and build on the geodesic sensor net that we originally created in the basement of Straub Hall. And many of you may be familiar with EGI. We had good success selling into research laboratories and eventually to clinical laboratories with the high density technique. And we eventually sold to Philips in 2017. And since then, BEL has become an independent company, carrying on our basic science kind of as our major focus, but also now building a new generation of products, and including not only our initial focus on source localization, but also mapping a complete high density EEG system. And I'll introduce that a little bit later. But the point of this webinar is to really go through basic principles, a few principles of how electrical fields are generated in the brain. And our goal really is to be able to teach anybody those basic principles so they can understand the value of EEG. Our assumption is that EEG is the premier in neuroimaging technology. The dynamic activity of the brain requires high temporal resolution that only MEG and EEG can provide. And with a number of advances, including precise special characterization of the conductivity of head tissues, EEG is now as good as MEG, and this is increasingly recognized. And I think we're also interested in transcranial electrical stimulation in the advances of a high density system. I'm not going to emphasize that today, but it really follows from a lot of the same principles. So here are three publications that I want to emphasize, and I'll include this in the slides that'll be available for you to follow up. But really comparing EEG to MEG, providing an overview of source imaging techniques, and then finally looking at the importance of high density EEG in clinical practice. The principles I want to emphasize today are fairly straightforward. And we're going to talk about the dipolar nature of the generation of the electrical fields. We're going to talk about the differential amplification, which leads to an important realization that it takes two electrodes to measure the EEG, and you can't ignore one of them, which is typically done. And it's remarkable how that error continues in the field today in current publications. Spatial sampling is a big obvious issue. Do you really need a lot of channels? And we'll look at when you do and when you don't. And finally, the way that the dipolar fields propagate through the tissue is through volume conduction. So we'll look at the principles of volume conduction and what that means for the importance of anatomy and really characterizing an individual's anatomy. And we'll do our illustrations within Sorcerer. This is the software that BEL has been developing for the last 5 years to allow high resolution EEG through including all the properties of the head tissues that allow source localization. So we'll use that as our illustration tool. But I want to emphasize that Sorcerer is not BEL's only product, that we've now built a complete high-density EEG system based on the geodesic head web, the next generation for high-resolution sampling at the head surface. So I think the point is not only the EEG is very powerful, and by first principles is the best neuroimaging technology, but also it's increasingly affordable with systems like the new BEL System 1 that we're introducing. So let me start with the basics, the generation of the EEG. Many of you are familiar with this, but I think, again, we want to make this an introduction so that every new lab member can have a set of principles to understand and build up their knowledge from the basics. So the generation of the dipolar field from the cortex comes from largely the pyramidal neurons. And it requires a lot of these pyramidal neurons to become more or less synchronously active in order to generate a far field, a field that can be measured at surface electrodes. And you can see that the propagation of the nerves from the apical dendrites to the neuron soma is the primary generator with the return currents in the extracellular space. And it's dipolar. The nature of a field measured more than very close electrodes is only dipolar. The other poles of the electromagnetic field drop to zero. And that dipolar field is an essential principle to understand the propagation of the far fields as measured by the electrodes. Now, in this textbook example, there are a couple of errors. One, there's only one EEG electrode. And whereas indeed we need 2 electrodes because we use differential amplification. So the reference electrode not shown in this image is a priori half the signal. But also all of the neurons are lined up uniquely in a radial orientation to the cortical surface. And in reality, the human cortex is wrinkled. In fact, it's highly convoluted. And that means that we have to understand the wrinkles of the cortex to understand the orientation of the dipolar fields. So here I've shown a dipole in one region of the cortex. Here it's pointing negative up. So the electrode at the surface of the head nearest this cortical sheet will have a nice strong negative potential compared to a positive potential measured elsewhere, such as from a reference electrode. Now, the big circles illustrate the right-hand rule of the magnetic field. And so here, for this radial source, the electrical field is strong near the nearest electrode, but the magnetic field is 0, perpendicular to those right-hand rule circles you see. In contrast, if we put a source in the sulcus, in the wrinkle itself, that will be oriented tangentially, and the electrode at the top of the screen, in other words, the nearest scalp surface, will see a 0 potential, even though it's the closest electrode to the source. And that's because the tangential orientation puts the dipole with the negative field to the left and the positive field to the right. And here, as you can see, those right-hand rule circles are showing the magnetic field will be strong at the sensor closest to this electrode. So both the orientation and the dipolar field are really the critical first principles to understand. And as we measure these sources, we have to consider the orientation of, and the location of the reference electrode. And that's easily illustrated if we imagine a source here. So on the left, I put that blue square at a region in the brain. And on the right, I've made a surface characterization of the voltage fields that would be seen with a reference at infinity, or an independent reference. And what you can see is there's a positive field that we're illustrating in red, and a negative field right next to it, illustrated in blue. Now, if you put a reference electrode, and again, we're looking down on the top of the head with the nose at the top. If you put a reference electrode on the left mastoid, as this dipole becomes active and inactive, that variation will be zero over in the area that's white. But if you put a reference electrode on the right mastoid, suddenly the characterization, if you assume that's zero, your characterization would be highly wrong. And so as the dipole comes and goes, you can see that the reference electrode is changing its voltage depending upon where it is. And that's really true for a lot of well-known EEG features, including evoked potentials or event-related potentials like the P300. Depending upon where you put your reference electrode, the P300, or the positivity at about 300 milliseconds, is an N300. And in fact, that's what we see if we use a vertex reference electrode in that case. So understanding how those go together, it really makes a common error to label a channel with just one electrode. Indeed, every differential recording is probably best labeled with two electrodes, the reference and the so-called active or index electrode. Now, how do we avoid this differential recording and map voltage at the head surface? Because it's hard to map a differential. We need a zero reference if we're going to have a single monophasic mapping. And this has been known for a number of years, and that's to have enough electrodes, you can characterize the entire surface of the head. This is the concept of the zero surface integral from Bertrand and Associates. And it's pretty easy to understand. So for the example I showed here, if we map the potential all across the surface, it will add up to zero. And that's because the positive fields and the negative fields totally balance. And it doesn't matter how many dipoles you would have in the brain, the same principle still holds if you can measure the entire surface of the head, including estimating the cross-section at the neck, the integral would be zero. And that's why an average reference, if you have enough channels, becomes a good approximation of the head zero. Now, there are other more precise approximations, such as the polar average reference approximation that Junghofer and associates developed a number of years ago. But the average reference with a high density array gets pretty close to the zero integral, and that's because we have enough electrodes on the bottom of the head. And on the right, I'm showing the geodesic head web, the 280 channel version. And on the left, I'll show a conventional cap and where the electrodes are put on the top of the head. And by these first principles that have been known for many years now, that would not be the optimal measuring strategy. Those electrodes are close to the brain, but the brain's fields are propagated by volume conduction throughout the entire head volume. So the electrodes on the face and neck are actually essential to map the entire brain fields, particularly the basal surface of the brain, which is very important to almost anything you want to understand about brain physiology. So I thought it might be useful to do a kind of a real-time lab demonstration of dipolar fields using the software. sorcerer that we do for mapping. So let me stop sharing here and ask Wei to step in and give an illustration of mapping fields from the cortex, measuring them at the head surface with a forward projection in Sorcerer.
Sure, thanks, Don. Can everyone see my screen?
Yep.
Okay, so one of the features that Sorcerer offer is this estimate source from forward projection. So this is the head model. On the right here is the voxel head model, the dipole models, a 3D dipole model. So let me pick a source. Let's see, from the a deeper region, maybe in the middle here. Okay, so let me toggle the orientation of the dipole so that we can better. So now I'm selecting a dipole that is in the dorsal DIPFC area. As you can see, the orientation is projecting. Oh, actually, this is in the more frontal. Let me find a different one. Sorry. Okay, here's a better one. Choose another one. As you can see, the dipole here, projection, this is a ventral anterior singular. The projection here on the top, the parietal area is kind of a whitish, and this means that the voltage on the top here is close to 0. Now, according to what Don said, that usually indicating where the source is beneath. But this is actually, source is much deeper than actually this power projection here shows. So this is based on the CZ. Now, if I change that to a average referenced data, now you can see the projection actually have a much stronger positivity on the parietal area. And indeed, increase the opacity here, the close to 0 projection is more deeper, and it's more consistent with the actual depth of this source here, the dipole that I choose. Now you can see that that's why, you know, when you use the average of references versus individual references, that will give you a much more truthful information than the actual using individual references. So let me stop sharing and turn back to Don.
Yeah, really the goal here is to be able to illustrate these principles in kind of a dynamic framework. That's part of the design we did in building Sorcerer, as much as a teaching goal as just computing source localization. Another principle is the spatial sampling. How many channels do you need? Could you just use a few channels, or do you need a dense array? And there's a long tradition of work. I've listed a number of publications in the slides that have guided our understanding for many years, starting back with the textbook on bioelectromagnetism by Malmivwo and Plonzi, but then follow-up applications specifically to the density of spatial frequency in EEG and MEG by Malmivwo and associates in Finland. And fundamentally, you need a lot of electrodes, and yet it's asymptotic that if you have fewer than 64, it's really hard to do accurate source localization at all. But if you have 500, you then capture the great majority of the information. But of course, that's challenging practically. To illustrate the aliasing that can happen with too few electrodes, we can look at aliasing in the temporal domain. So what I've basically done here is to have a signal shown at the bottom of the screen in white, and look at the digital characterization with various rates or frequencies of digital sampling. And you can see if we get close to the frequency of interest on the left with our digital sampling, we do capture the dominant frequency of the oscillation. On the right, however, we've cut down the digitization rate, or the number of samples per second, and suddenly we alias. The recording looks as if it's half the frequency of the original signal. And this is well known in the temporal domain, but it's not so well known in the spatial domain. And over a number of years, we have tried to characterize the principles of spatial sampling using the basic Nyquist frequency. And now, instead of looking at one dimension, we really have the dimensions on a sphere, or the head surface, that we can characterize with radial coordinates. So the azimuth and the altitude from astronomy is a good way to look at the radial coordinates. And we can use that to artificially describe spatial frequency. And so in this paper we did 20 years ago or more, the low spatial frequency in the top row is fairly well characterized down to 32 channels. And so now that low frequency would be as if one lobe of the brain is active. Now, if you have more regional specificity, or a tighter focus than just one lobe of the brain, then you can see how the spatial frequency is mischaracterized, even with 64 channels. You cannot reconstruct the entire spatial frequency. And even these are fairly large gross areas of the brain that are active. So the spatial frequency really depends on how much of the brain is active at one time. And given the kind of anatomical specificity of the brain, even large synchronous activations are often at a high spatial frequency and can be missed with an array that's inadequate. One of the things we did soon after the theoretical work is to look at clinical signals. And so here we have a delta frequency, or a EEG slowing after a stroke. And it's a fairly big area that was stroked in this patient, the left or the right posterior, using a neurological convention of this image. And what you see is that it's characterized fairly well, even down to 32 channels. But at 19 channels, we see spatial aliasing. The regularization of the voltage at the head surface is actually incorrect because of too few channels. And one of the things that we've learned from a number of our associates over the years is that kids' skulls are even more conductive than adults. And so, particularly for infants, you need more electrodes than you need for adults, which of course is not the clinical sense, because many times babies are only measured with 2 electrodes, but they actually have higher spatial frequency that's required. So let me stop at this point and ask Justin maybe to give us another example of spatial sampling and what it looks like when we can generate an artificial source and look at its effect in sorcerer.
Yes, thank you, Don. Hello, let me share my screen. I'm sharing here from Dallas, Texas. Hello. And you should be able to see my screen here. All right, and as Don mentioned, it's of interest, of course, to see a difference between subsampling, higher sampling, 19 channels, 280 channels. And one of the tools that we've developed is actually a combination of simulated data, 280 channels with various frequency bands and event-related potentials built into the data set. And so if I go into this sorcerer window here in my Chrome browser. I will pull up a data set that has an event-related potential located to the left occipital cortex. And I'll show you a source. I'll click compute here. I'll show you a source. You can see in the upper left-hand corner, a cortex overlaid with 280 sensors, EEG sensors. Here you can see a flat map, an expanded, inflated brain. And in the right, in a butterfly plot, you can see an overlay of these composed signals. And at time 0, there's an event-related potential that would be localized to the lateral occipital cortex here. And you can see this right here. You can see this nice localization. And to give you an example of what this might look like if we had sampling at fewer channels, say 19, I'll enter this here and compute. And you see this much more broad, very diffuse region of localization. You see the evoked potentials here, but it really highlights the focal accuracy that we can get from higher channel counts. And if I show a few slides on this, it becomes apparent. So here we are at 19 channels and then in at 280. And we can do the same thing with a source in the right medial prefrontal cortex, right? And again, 280 channels versus 19 channels. Quite a difference. And on that note, I will hand it back to Don.
Good deal. Thanks for that. That's a good illustration. And what I think it's really interesting to do is to vary the channel density across a range and look at the increased precision with each channel density. And it depends on the source. One of the general principles is a deep source projects somewhat diffusely to the head surface. And whereas a superficial source has a very tight field that absolutely can be missed unless you have high spatial sampling. So many epileptic spikes we see, even with the 19 channel array, as long as they're deep in limbic regions. Now, we can't localize them that well, but they are clearly visible. But some very superficial cortical spikes can be between the electrodes and actually missed with a conventional clinical array. So really the projections that Wei and Justin have been showing you are because we build a precise head model. And that means we map the volume conduction or the propagation of the electrical fields through the actual conductivity of the head tissues, where the skull is the most resistive field. So we need to map not only the dipolar fields and their orientation with the cortical surface, but also we need to consider the conductivity of each tissue. Now, in this slide, I'm going to mention kind of a recent discovery that we're very excited about. For many years, we thought we could achieve precise source localization only when we had the individual's MRI. When we use an atlas models, particularly with the triples, as we've been illustrating, the precision is not very good. And it was because we didn't have oriented sources, and therefore we had to compute all three orientations, the X, Y, and Z orientation fields for every voxel. And that's just not that good, actually. What we've discovered recently, and we're now working to put in the next release of Sorcerer, is that we can compute an average orientation from the Montreal Brain Atlas, the average cortex. And it's not so bad when we compare it with an individual. So we're very excited to get our Atlas models upgraded now for the orientation, the average brain orientation. And we've got more validation to do to map it against individual data, but we think this will be a big step forward in inexpensive neuroimaging, where you don't even need the individual's MRI to have an accurate source localization. So as we've been discussing, the forward problem is how the electrical fields of the brain get out to the head surface, where they're measured by our electrodes. And the inverse problem is, how do we take the data at the electrodes and figure out what generated it in the brain? And you can see in our example here that the cortex is highly wrinkled. And so every little dot that we use as a dipolar field there really depends upon the orientation of the cortex at that region. So it really matters to understand that orientation. So for many years, we have been trying to work out these first principles. And a lot of this work was done at the University of Oregon Neuroinformatics Center, where we looked at tissue properties, estimated the conductivity through each of the tissues, and then worked out a technique for using that conductivity in the computation of the source modeling. And this process was helped a lot by Kai Lee, a PhD student at the University of Oregon, who then helped us to develop a complete segmenting workflow, so we can characterize each of the tissues that matters. So we have 7 tissues, including not only gray and white matter in the skull, but the cerebral spinal fluid, which actually has a big effect on the propagation to the skull. But also eyeballs and air and scalp, they all have to be characterized accurately so that the relationship between each electrode and the underlying cortex can be specified in the mathematical model. That's really the forward model. And the most problematic tissue is the skull. So in every atlas model, we have a skull, and we characterize its conductivity from the known properties. Also, for individual models, even though we don't get a skull CT, we warp a atlas CT, so we have a skull model for each individual. Within Sorcerer, we build those tissue properties, organized on Flow, which is our laboratory data information system. So I thought now maybe I'd ask Viral to give us an illustration of how we use the tissue properties and how we visualize those in Sorcerer.
All right, can you guys hear me okay?
Yep.
Awesome. So what I wanted to cover today is to look at head tissue conductivities with the MRI visualization tools and the different choices for how to look at the MRI and how to relate that then to the head voltage fields. So to illustrate that, I've loaded an epilepsy data file here in Sorcerer software. So Wei and Justin has already provided a nice overview of the 2 3D views that you see on the top. But what I wanted to draw your attention to today is the 3 2D views that you have at the bottom here. And so what we've done here is we've segmented the brain into 7 different tissue types. And these are MRI slices in X, Y, and Z direction at the bottom. And these 7 different tissue types are characterized by different colors. And so, for example, you have yellow at the very top that points to scalp, purple here that points to skull. You have CSF here in cyan, you have gray matter, white matter, you have dipole seated in the gray matter, as you could see there with those voxels. You also have eyeballs here, as you could see, and so on. So you could manipulate and view the slices in various different ways, slice by slice, however you want. There are also, you could view that as a single slice in a very large view if you would like. And within Sorcerer, just to kind of give you some additional perspective, we have various age appropriate atlas models. all the way from infants, 0 to 2-year-old, through adults. And so, again, going back to sort of the data file that we're looking at here, here's the epilepsy spike that you see at the bottom right in the butterfly plot. So I'm localizing halfway up the spike here. And you could see the forward voltage projection showing posterior temporal occipital orientation. And if I were to snap to maximum here, you see the M and I coordinates. If you want to try to look at it in a very standardized way of estimating your sources. So you see it's in the fusiform gyrus here. And one last thing that I'll point to is this is also a very nice view in case you're looking, for example, as we release the next individual head models in our software in the future. This particular view here, for example, if you have lesions within the brain, you'll be able to see whether your source estimates wind up in the lesion or not, and things of that nature, the top right 3D volume model that you see. So with that, I'll pass it back to Don.
Great. Thanks, Viral. I think that's a really nice illustration from the epilepsy data. And actually, what I'll do now is to give some examples from our work with epilepsy. It's been an area where we can validate our source localizations against intracranial recordings and intracranial lesions. So our first work was done at University of Washington with Mark Holmes and Jeff Ogeman with pediatric cases. And this is back when the highest density we could get was 128 electrodes, and our source localization was GeoSource 2.0, a fairly simple first effort that we weren't sure was going to be that accurate. But it turned out to be fairly reasonable with the 128 channel data to localize the onset of the epileptic spike and seizures. So we then made a prediction of where we would expect the intracranial recording to show the feature. And that was in the red dots here, the occipital lobe. Now, if I just go back, you can see the left occipital pole is where the non-invasive mapping fairly consistently showed it in this young patient. And the intracranial electrodes, in this case, strips and grids, were placed, and the red dots show the onset, the very beginning of the seizure, in the lower left-hand traces. And you can see the signal there is a gamma oscillation, a high-frequency oscillation right at the seizure onset that then generates lower frequency oscillations. You can see the bigger waves, and those are actually what we can localize. Right now, the high-frequency oscillations are tricky to localize from the scalp electrodes, but fortunately, we can localize the lower frequency features that rapidly follow. We've been able to actually do simultaneous high-density recordings with intracranial electrodes with our colleagues in Japan at Seirei Hamamatsu Hospital. And the procedure is fairly straightforward. We wait for the wound to heal of the exit of the wires from the intracranial recording, and then we can apply the high-density array without worry about infection risk, and then compare the same events, epileptiform discharges, intracranial and extracranial. And this is actually a good illustration of the basic principles we've been trying to go through, because what you see by an electrode that's right next to the generator, here it's a right anterior medial temporal, illustrated by the yellow electrodes on the lower left. It's a big spike when you're right next to it. At the head surface on the right, however, it's not so big. It still has a distribution that is analyzable, but we have to watch the noise levels of the recording, and even visual inspections often somewhat challenging from the surface. So there is an attenuation primarily through the skull, but through all of the tissues. Nonetheless, the localization has been really quite good from the non-invasive scalp electrodes as validated by the intracranial recordings. Now, the other validation that we do, and this is now a recent paper using sorcerers, multiple sparse prior are really our best inverse technique from Washan Hospital in China. Here we use the surgical outcome. Can we predict what the neurosurgeon's resection will do to make the patient seizure free? And here it actually turns out that the non-invasive mapping is a good guide to the neurosurgical, even without the intracranial electrodes. Finally, I'll give another illustration of a recent publication where we're localizing the oscillatory components. This is a wavelet analysis to characterize frequency features of the EEG, which we localize directly. So here we're not just localizing power, but we're localizing the power at a specific frequency. And that actually reduces the noise of the source localization and is very useful in characterizing the very beginning onset of the seizure. So part of our development is to integrate intracranial recordings, which you see in this image in Sorcerer on the upper left as the tracks of depth electrodes inserted through holes in the skull into the brain, and to integrate the data from the depth electrodes with Sorcerer at that point. So at this point, I'll stop, and that kind of concludes our lecture, and maybe we can go through question and answer for a little bit.
So the first question we have is, do we take into account differential tissue characteristics for electrodes placed on the face and neck?
Yes, that all of the tissues in the head surface are important. The skull is the most important because it's the most resistive. But also in the face, those face electrodes are pretty far from the brain. They do capture features that you don't see unless you had electrodes there. We often have an anterior temporal lobe spike that's not easily visible except on the cheek, because the spike is pointing to that region. And here, all of the air in the behind the nose and all the sinuses are important. So those are indeed modeled in Sorcerer.
Okay, and then the next question is about the channels of electrodes. If we increase channels from 128 to 256 channels, how much will it affect source localization accuracy?
And here it really depends upon the sources. As I was saying earlier, if we have a deep source, it's fairly well characterized by the 128 channels. So we do see an increase in accuracy. Now, I don't have good parameters for that. You know, I would say roughly, we think that 2 to 3 centimeters is pretty accurate with the 128 array. And with 256, we think we can go lower, maybe even to 1 centimeter, or with the 280 array, which is our current high density. But I would emphasize it's asymptotic. So the 64 is probably enough to do source localization. A 128 or a 130 channel array is actually the sweet spot if you are really limited in the lab budget for reasonable source localization. But the improvement up to 280 channels is really considerable. In fact, we're looking at developing higher channel counts, 400 or 500 channels, just because we've never really known the practical accuracy until we did the studies. And we think those will be necessary at the next step.
Great. The next question is, to what extent do you include volume conduction effects in non-cephalic electrodes?
Yeah, very good question. Very similar to the other one. Those are as equally important. So the scalp, the neck, all of those tissues are associated with conductivity in the three-dimensional model in Sorcerer. So they're part of the forward projection. And as I mentioned, the air is a big resistive region up in the front around the face. And it will limit the forward projection, and therefore needs to be considered in the inverse projection as well.
Okay. Next question is, the maxilla and hard palate included in your head models?
They are considered as flesh. So in our 7 tissue compartments, as Viral was illustrating, showing the visualization of each of those tissues, it's not a fine discrimination. There are actually some finer discriminations of the conductivity of each of the tissues. And right now we don't have those. And I don't think those are visualized. Actually, I'm not totally sure, but I don't think they would be in the CT skull image. So any bone that shows up in the CT is assigned to bone conductivity, but I don't think those particular tissues are actually.
Okay. Next question. Is it possible to localize 2 sources in one EEG session? For example, one from auditory P1 aiming lower auditory cortex and one from auditory P2 aiming higher auditory cortex?
Sure. It just naturally follows out that you can source localize different features from different times in the same model, in the same head model. They're just 2 source localizations. And I think furthermore, if you wanted to analyze the interaction between these, that could be done in the same session too, particularly if you've mapped somebody carefully and you knew their basic sources for P1 and P2, you might be able to then look at individual trials and study the interaction, starting with those prior sources. So that's actually a very interesting research approach, to start with basic localization and then build up more of the dynamics within the experiment.
Great. Next question is about the electrodes, and I would try and get the phrasing right here. Is not too short a distance between the electrodes carry a risk of short circuiting them? Very
good point. We worry about this in terms of bridging between electrodes. And basically with the most typical electrolyte that we use, which is a saline sponge electrolyte, when we first put the head web on, there will be shunting between the electrodes because there's saline that bridges. But within 3 minutes, 5 minutes, that dries out. And we can measure the bridging in the software electrically. We can assess the degree of bridging, and it very quickly dries out. Now, with a gel electrolyte, we have to be more careful. If the head web is mushed around, you can get bridging, which then will affect the signals. Now, the interesting thing is the short circuiting or bridging is not disastrous. It's just one more thing to account in the conductivity model. And it doesn't actually affect the potentials as much as you might think. In fact, we've even thought about strategies of covering the whole head with electrolyte and then putting the electrodes on. But you do have to put it into the mathematical model because with the volume conduction act through the skull, it hits that area and then conducts very rapidly between the electrodes.
Don, can I also add in that the open structure of the net makes the evaporation much faster? Would you agree with that, as opposed to something which is more of a closed surface or closed?
That's right. That's right. No, that's a very good point. And the other thing is that when we use the gel electrolyte, for example, for long-term monitoring in the epilepsy unit, the open structure of the head web allows us to see the bridges. We actually can inspect the area. So it's better than a cap that you can't see what's going on underneath it.
And here's another question. Can we use two 126 channel caps together with a single 256 amplifier for recording simultaneously from two subjects?
Yes, very definitely. Basically, what we do is we synchronize the data acquisition across each of the amps. So you might have a 130 bell amp and a 280 bell amp. And by sharing a TTL synchronization between them, they can be acquiring with millisecond accuracy. Now, in that case, each would acquire its own data file, but with the precise timing, you can then merge that and do hyperscanning or across individual mapping with that technique.
And there are
Great.
some nice publications on that if folks wanted, based on some of our prior experience with the EGI platform as well, in terms of hyperscanning with multiple participants.
That's right. A number of labs have done that. Yeah, and it's really very important for understanding social interaction and communication. It is challenging. It takes extra work to do the cross-participant data analysis primarily, but the acquisition and the timing is fairly straightforward.
Okay, we have a question from a rather lengthy question in the chat. This is from Erica Flayton. I ran an oddball listening task with adults using the 128 geodesic net. I'm interested in using source analysis, not to locate the exact sources per se, but to use as a spatial filter for comparing mismatch negativity for different conditions. I do not have the unique electrode positions for each participant, so I was planning on using the grand average MMN to fit the dipoles to a validated generic head model to try to get auditory cortex generally. Then I was going to use the MM peak of the individuals to fit the dipole orientations individually. This way, I use the same dipole location, but different orientations to get the source wave for each condition I'm interested in for each participant, and then compare the conditions. Does this make sense?
It's a very creative approach. I'd have to think about this one. I know what you're trying to do. You're trying to use the orientation from the individual to then improve the measure or the mismatch negativity amplitude estimation. It sounds good. It sounds like the logic is very good. Practically, I don't know. I'm going to reserve judgment and until I would get a chance to see the data and go through it. But the logic is good. Basically, to start with the generic localization and use the orientation to then recompute each individual signal as a function of it. I'm just not sure how that would work. So maybe we could follow up offline with that question. It sounds very interesting.
Sounds good. And we just have one other question I can see right now. Can you use electrical distance to check for bridging?
Yes. In fact, that's the way we do it, which is just the signal identity. So even two closely adjacent channels will have some difference in the signal. And if they don't, they have enough difference, then we will suspect bridging. And the way we validate it is we create an artificial bridge and see if that's true. Now, it's an interesting thing that the bridging, if you put a bridge between two channels, they're not exactly identical because they still have their own projection fields, but practically they get a lot more similar. So it's easy to set a threshold for bridging, and practically validate, and which is what we do. And our theory isn't quite that precise at that close level. So we make a bridge, evaluate the similarity of the signals compared to when there's no bridge, and set a kind of empirical threshold. But that is the logic. Yep.
Great. Well, I think that wraps up the Q&A portion of our webinar. And just to let everyone know, we are going to be putting this recording online on our website, so you can review the webinar at your leisure or send it to colleagues. And I want to thank you very much for everyone for joining this morning, and thanks to all our speakers as well.
And I'll add, we're very interested in these questions. So if you send us a note, we're very glad to follow up with those, whatever advice and help we can provide.
Great, thank you.